Thank you for the opportunity to share the work that I've been doing as part of my PhD project on antigenic refocusing of a SAT2 virus. Foot and mouth disease virus is associated with antigenic variation. Therefore, it resulted in occurrence of multiple subtypes within the SAT2 serotype. Foot and mouth disease virus induces strain-specific immunity. Therefore, there's poor cross-protection between viruses within the same serotype. Problematic pathogens like HIV, influenza virus, including FMDV, evade the immune response by inducing strain-specific immunity, and this mechanism is known as deceptive imprinting. These imprints are surface-exposed, hypervariable immunologic regions known as decoy epitope. There's a strategy to prevent deceptive imprinting, and this strategy is known as immune refocusing. This is simply done by masking or removing these decoy epitopes. So towards immune refocusing of SAT2 viruses, we aim to identify the antigenic determinants of those viruses by charge dampening and replacement of these hypervariable regions. To construct the mutants, we use the SAT2 ZIM7N3 full genome infectious clone, and we did that by replacing the P1 region with the P1 with desired mutation. We constructed 10 replaced mutants by swapping the corresponding regions of SAT2 ZIM7N3 with that of antigenic distant SAT2 Egypt 9-2012. So of the 10 mutants, the first one, the first one we constructed a combination of two epitopes to make a single mutant in VP3, and then we had 4 mutants where we replaced single mutation in VP1, and then we had 3 mutants where we replace either GH, C-terminus, or a combination of the two. And then we also constructed 2 chimera viruses who replaced the complete P1 region to that of Egypt. And then on the last chimera virus here, we replaced the GH loop back to ZIM7N3. So the 3 surface exposed proteins, they are from Egypt virus, and then the GH loop is from ZIM. Then we also use the charge dampening strategy where we replace the epitopes with alanine residues, and then for that we had 3 mutants where we replaced 3 epitopes in VP2, and then 1 mutant where we replaced single sites to alanine in VP3, and then we also had 4 mutants where we replaced 3 regions in VP1 through alanines. So the first thing that we wanted to see, is to see the impact of these mutations on the phenotype of the viruses. So we did plaque morphology in BHK, in IBRS, in ZZR, and CHOK1 cells. And for all the alanine replaced mutants, we found that the phenotype was still the same. But then, for the chimera virus, we found that they produce smaller plaques compared to the VSAT2, which is the ZIM7N3 infectious, the clone. And then in CHOK1 cells, we found that the CT mutant and the chimera viruses produce microplaques, and these were only visible after 22 hours of incubation, while the GHCT and the chimera viruses with ZIM GH loop, they could not infect the CHOK1 cells. Then we went on and used virus neutralization tests to test the antigenic distance of these viruses when we compared to VSAT2, and we did that. We used homologous sera, ZIM7N3, and heterologous SAR304. In this case, we used SAR304 instead of the Egypt sera because we didn't have that, and then because SAR304 has a broader neutralizing activity. For all the alanine scans, they show similar reactivity to SAT2. But then with the Camara viruses, we found that there was poor cross-reactivity towards both sera, and then even when we replaced the GH loop back to that of ZIM783, there was not much difference in reactivity. And then with the CT mutant, we found that there was a reduction in neutralizing titer against both sera, and then also similarly with the GHCT, there was a reduction in reactivity only to ZIM783 sera. So these results confirm that the GH and the C terminals, they are indeed immunogenic. And then with the replacement of region 46 to 50 in VP1, the SAT3 mutant, we found that there was a reduction in reactivity to both sera, and then we think that this site here might play a role of a decoy epitope. Then we went on and did antigenic profiling of these mutants against the SAT2 specific monoclonal antibodies, courtesy of Pebblad Institute. We use VSAT2 as a point of reference with 100% binding, and the value that I shown here is the percentage binding towards each monoclonal. With the Camara virus, we found that there was a reduction in binding towards G1, G11, and G12. And then with the same mutants that we found a reduction in reactivity to ZIM783 and SAR03 sera in virus nutrition tests, we found that there was a reduction in binding towards DA10. To bring your attention to these two mutants here, we found that there was a reduction in binding towards 1G5, G1, G11, DA10, and this result confirms the finding that we previously found in the work that was published by Opperman 2014, the work that was done in our lab. And these results confirm that these two regions, BP270 to 71, BP2 132 to 133, are indeed the antigenic regions of a SAT2 virus. And also in agreement with the same study, we found that there was a reduction in binding of EHER mutants where we replaced this region, BP1, 81 to 87, to alanines. There was a reduction in binding towards all the monoclonals. The results that the Opperman et al. Study found is different from what we found here, because they found that there was a reduction in binding of the SAT3 mutant towards 1G5 and G11. So, but I will explain to you why we got different results here. And then lastly, with the VP3 mutant, we found that there was a reduction in binding to all of the monoclonal antibodies. And this mutant here produced very small plaques, and it was always difficult to get high titer. So we're not sure if this result is because of the test. So we still need to do more work to validate these results. Okay, here is the two regions that I explained that we found reduction in binding towards 4 monoclonal antibodies. So this region here, region 71 to 72, and region 132 to 133 in VP2, where confirms that these two regions here, they are antigenic sites of this virus. And then when we replaced region 83 to 84, we found that there was also reduction in binding. And then, but when we replaced this region 46 to 47, we were only able to change the arginine to alanine, but the threonine we couldn't change. So the threonine sits here. So we think that's why we didn't get any reduction in binding. So in conclusion, replacement of VP1 C-terminus in combination with GH loop resulted in reduction in nitrogen titer, showing the significance of this region in immunogenicity. And then region 43 to 50 in VP1 seems to play a role of a decoy epitope. And this work also confirms that residues 72 to 71 and 132 to 133 in VP2 and residues 82 to 85 of VP1 are antigenic side of SAT2. Thank you.
Thank you very much. Do we have a question? No? Okay. Thank you very much. So I think then we have one